All right, so we're going to look at gene regulation. That is regulating the genes. What does that really mean? It's whether it's on or off, whether that gene is being transcribed or not. So gene regulation, it's just several different processes that are going to determine if a gene is going to be transcribed, end up forming a protein. And remember, those proteins can be hormones, enzymes, as well as our traits, which the enzymes help make. So think about it. If we had all of our genes on, every single cell in your body has all of the DNA, 46 chromosomes, except for your sex cells. That means the cells in your cheek have every single gene to make your entire body. The heart, the lung, lips, nose, anal sphincter, all of that stuff is here. But thankfully, all of those are turned off except for the genes that we need to make a cheek cell. Likewise, our tongue, all of those genes are turned off except the ones that make it a tongue and what it needs. Okay. So this gene regulation is determined by three different things. Number one, the need to maintain homeostasis. If we're producing too much of a particular hormone, the body's going to turn that gene off temporarily to get that back into levels. If your hormone levels are off, you're out of homeostasis. I'm out of homeostasis. I have a thyroid. It doesn't produce enough hormone. That gene permanently turned off. So I have to actually take supplemental hormones every single day to make sure that my body is getting that hormone. So one, do we need it? If we need it, that gene's going to be turned on. If we don't need it, the gene is going to be turned off. Secondly, we need genes turned on and off to make specialized cells. Our cheek is a specialized cell. Our tongue, our eye, our heart, our muscles, our skin, they're all specialized. But in the very beginning, you have 23 chromosomes from your mom in the sperm, 23 chromosomes in the egg from your mom. When they unite, that forms a zygote, which is a stem cell. That means all of those genes are on, every single one. And then probably about 4 or 5 days later after conception, you have cells starting, and they're starting to differentiate, become different, become specialized into skin, into bones, into brain, into heart cells. So genes are being turned on and off in those particular cells. So 9 months later, you have a baby that has trillions of cells, of which they are highly specialized. We only have a few cells that remain as stem cells. Those that produce our blood, right? One blood stem cell can produce white blood cells, red blood cells, and the different types of white blood cells. So specializing cells have certain genes turned on and off. An example in a plant, I'll run to the back. When you're looking at a stem, are the genes turned on for roots? Do you see roots coming out of the stem?
Yeah. Oh, wait, no, not on the stem. On the stem.
Are there any roots here on the stem?
No.
There are no roots. But listen, when you take off a stem from various plants, like I did from this milkweed plant, I stuck it in some water, and lo and behold, the gene, I turned on the gene by placing it in a different environment for those roots to come on. Now I can plant this and make some money. So off of one plant, you can cut off the stems, place them in water, they'll root, then you can plant them, and you can make how many plants off of one? So changing the environment. The next thing that determines whether genes are turned off is our environment. Our nutrition. What is our diet? That turns on a lot of bad genes depending on what you're eating, or turns off a lot of bad genes. The exercise, are you exercising? Exactly. If you're not exercising, genes go off, certain genes go on. Which ones go off are the good genes. What's on are the bad genes. And then finally, our environment. Stress, chemicals, radiation, all of those can also cause our genes to come on and off. So that is gene regulation in a nutshell. When we're looking specifically, there are 3 different ways. We're going to look at one. There's pre-transcriptional, right, turning on and off before it's even transcribed. After the mRNA is made, that's kind of spliced and rearranged. We're going to focus on this one. Epi, like your epidermis, your skin, that means above. So it's above your genes. Epigenetics, all gene regulation, does not change your genes. It just is going to either turn it on so it could be transcribed or have it turned off so it can't. So it's all about changing those, whether the genes are expressed. If you eat foods or taken a chemical, methyl, right, that actually puts a little tag just on the cytosine. Right? A goes to T, C, cytosine goes with G. That chemical actually attaches and it will block the RNA polymerase. So no mRNA can be made. No mRNA, no protein synthesis, no protein. Right? Those are bad genes. That's fantastic. But if they're good genes, that's a bad thing. Most genes that are turned off are good genes that we need. Another chemical, so this is actually on DNA, is called an acetyl tag. So if you eat food high in acetyl, garlic, onions, variety of other foods, this actually unwinds the DNA. So when this acetyl tag gets on the DNA, it unwinds it.
So is that a good thing, no?
I'll answer in a second. So when genes are unwound, yeah, the genes can be transcribed. Now the messenger RNA can be made, and the RNA polymerase enzyme here can actually get to this gene and transcribe it. Over here, the DNA is wound too tight. So no acetyl tags. If it's wound so tight, this RNA polymerase cannot get in there and actually transcribe that particular gene. So it's flipped. But this one is just unwinding the DNA or having it stay wound up. This one is actually attaching to the DNA. Okay. And there are special proteins that the acetyl tags will attach to, but that's beyond what we're looking at right now. Okay.